mechanism, activation and differentiation. B-cell activation is a critical step in the adaptive immune response and can occur through two main pathways. 1. T-dependent B-cell activation, most common for protein antigens. This mechanism requires help from T helper cells, specifically T follicular helper cells. It involves several steps: antigen binding and internalization. A naive B-cell recognizes and binds to its specific antigen via its BCR. The B-cell then internalizes the antigen through receptor-mediated endocytosis. Antigen processing and presentation. Inside the B-cell, the antigen is processed into peptide fragments. These fragments are then loaded onto MHC class II molecules and presented on the B-cell's surface. T-cell recognition and co-stimulation. An activated T helper, Th, cell, which has previously been activated by the same antigen, often presented by a dendritic cell, recognizes the antigen MHC class II complex on the B-cell surface. This recognition provides the first signal for B-cell activation. Co-stimulatory signals. For full activation, the B-cell also needs co-stimulatory signals. The most important is the interaction between CD40 on the B-cell and CD40L, CD154, on the T helper cell. The T helper cell also secretes cytokines, e.g., IL-4, Illinois 5, Illinois 6, that further promote B-cell proliferation and differentiation. Proliferation and differentiation. With both signals, antigen binding and T-cell help, the activated B-cell proliferates rapidly, forming a clone of identical B-cells. These B-cells then differentiate into plasma cells, which are specialized to secrete large amounts of antibodies. Memory B-cells, which persist for long-term immunity. Germinal center reaction. For protein antigens, this process often occurs in specialized structures within secondary lymphoid organs called germinal centers. Here, B-cells undergo somatic hypermutation, introducing mutations into the antibody genes to increase affinity for the antigen, and class switch recombination, changing the antibody isotype from IgM, IgD to IgG, IgA, or IgE to diversify effector functions. This leads to the production of high-affinity, class-switched antibodies. 2. T-independent B-cell activation for non-protein antigens like polysaccharides. Some antigens, particularly highly repetitive structures like polysaccharides on bacterial cell walls, can activate B-cells without T-cell help. This occurs through two types. Ti1 antigens. These antigens, e.g., LPS, directly activate B-cells by binding to pattern recognition receptors like toll-like receptors, TLRs, on the B-cell, in addition to binding the BCR. This can lead to a general, polyclonal activation of B-cells, regardless of their antigen specificity, at high concentrations. At lower concentrations, it can provide a co-stimulatory signal to antigen-specific B-cells. Ti2 antigens. These are highly repetitive antigens, e.g., bacterial capsular polysaccharides, that can cross-link multiple BCRs on the B-cell surface, providing a strong enough signal for activation without T-cell help. The antibodies produced are typically IgM and lack affinity maturation and memory. B-cell function. The primary function of B-cells is to produce and secrete antibodies, which mediate humoral immunity. Antibodies perform several crucial roles in fighting infections and maintaining immune homeostasis. Neutralization. Antibodies bind directly to pathogens, viruses, bacteria, or toxins, preventing them from interacting with host cells and causing damage. For example, antibodies can block viruses from entering cells or neutralize bacterial toxins. Opsonization. Antibodies coat the surface of pathogens, marking them for phagocytosis by professional phagocytes like macrophages and neutrophils. The Fc region of the antibody binds to Fc receptors on phagocytes, facilitating engulfment and destruction. Complement activation. Antibodies can activate the complement system, a cascade of proteins that can directly lyse pathogens, membrane attack complex formation, enhance opsonization, and attract immune cells to the site of infection. Antibody-dependent cell-mediated cytotoxicity, ADCC. Antibodies can coat infected cells or tumor cells, which are then recognized by natural killer, NK, cells via their Fc receptors. The NK cells then release cytotoxic substances that kill the target cell. Immune memory. Through the generation of memory B cells provide long-lasting immunity. Upon re-exposure to the same pathogen, memory B cells rapidly differentiate into plasma cells and produce large quantities of high-affinity antibodies, leading to a faster and more robust secondary immune response that often prevents symptomatic illness. Antigen presentation. While their main role is antibody production, B cells can also act as antigen-presenting cells, APCs, to T helper cells, especially during T-dependent activation. By efficiently internalizing and presenting specific antigens, they play a role in shaping the T-cell response.